Okay, so today we are starting a new beginning for Penn State, the One Health Microbiome Center, and the Microbiome Sciences. I'd like to introduce you to why we're gathered here today especially. So friends, colleagues, Professor Bosch, welcome. The Microbiome Medal Award, established this year by the One Health Microbiome Center, honors a singular scholar or a group of scholars who nobly extend excellence, acumen, and ingenuity in research, mentorship, and service to the global field of microbiome science. Today and forward, the medal recognizes scholars who not only advance knowledge and training, but importantly, they enrich humanity and they improve the lives of many people, especially ones they may never have met. They must motivate and accelerate the next generation of microbiome scientists. So it's a distinct privilege that the inaugural 2025 recipient is Dr. and Professor Thomas Bosch, who has and is playing one of the most formative roles in our community. As a thought leader, as an experimental maestro in illuminating how microbes shape host development, evolution, neurobiology, immortality, and cancer, some of which we'll hear about today and some of which we heard about yesterday. The author of over 180 papers in 3 books, a German National Academy Sciences member, which was established in 1652, a SIFAR Fellow, which is a think tank from Canada, the chair of Interdisciplinary Center for Applied Life Sciences at his home institution, Kiel University, the founder of the Center on Origin and Function of Metaorganisms, which many in our field have followed and have actually tried to reconstruct in the centers that we built, and a prestigious senior research professor at Kiel University. Professor Bosch continues to be a foundation, a catalyst in the development of the concept and literature on holobiont biology and metaorganisms, which essentially synthesizes hosts and their microbiomes into structural units and functional units that are in the hierarchy of biological life and its organization. Time and again, I have found, and many have found, that Thomas says yes when he's called into action for service across the globe. Time and again, he's invited. What did you say there?
It's a mistake.
It's a mistake. That's why you're here. So it's not a mistake. Time and again, he's invited countless microbiome scientists early in their careers, me included, to Germany to have spent some time at his center building international relations and reputations. I first had the experience to go there 12 or 13 years ago, and here we are today. And Thomas has always been promoting the young scientists since then. Time and again, he looks to learn with deep curiosity and inquisitiveness. And for those that he gets a chance to meet, you'll see that abundantly. And time and again, he engages the community to elevate the community. That's what this medal is about. Thomas, on behalf of the microbiome community at large, we are honored to bestow you the first recipient of the Microbiome Medallion, which really stands for a powerful and principled message that true academic leadership involves not only visionary discoveries, but the vital work of shaping the future of the scientific community through unparalleled service. I'd like to welcome you up to the middle of the floor, Thomas. If everybody could give Thomas a hand. Microbiome Medal, the first time anybody has seen it, and it'll hopefully be an annual tradition that you stand as a benchmark for as we go forward. Could you please open it and also show it to the community? Yes. Oh my God. Yes. So please show, and we'll leave the medal box up here so everybody can see it as they walk out. One final, what do you want a picture? Yes, yes.
No funny faces.
Okay, please, we'll look forward to your seminar. You're very welcome. One final thing is Benjamin Franklin said, well done is better than well said. Well done, sir. Okay. Yes.
Thank you very much, Seth, and the whole community. Somehow go down.
We made you tear up.
Yeah. To the honor of the day, I wear the socks of Penn State. They gave me yesterday. I'm greatly honored and deeply honored. It's a challenge to keep up, but also, honor which has a meaning, and I try to run this path as long as I can. And my very first word is going back to Seth, and he mentioned.
Need a little connection.
Maybe you just need to.
No, it's thing there. And
Hey, all right.
here you are, about
I
2016.
looked a little different then.
Yeah, you look different. You look better now. Says Budenstein, it was immediately clear that there's a new center point coming up and as a leader, and so we had a very good time. And I do on YouTube, you can find I do numerous interviews with key persons in the field, and so we had says here it's about 1,600 views already on this interview, but it just means this is common thinking, and the field is emerging, and the field has leaders which are driving the field in the new world. And the new world is a holistic world. I want to introduce the term think metaorganismically, think holistic, think in networks. That's where the new medicine, the new biology, the new evolutionary biology will be. Getting out, we talked about it earlier, get out of the silos, come together. I had a wonderful walk. Thanks, Marianne, from the hotel to here. And the campus lives that already. You have the theater next to the geology, next to the life sciences, next to something. And I think we have to do that also in our daily life and in our sciences. So thank you so much. It's a great honor, which has a very special place in my life. We, the bacteria, makes no sense to you. If you see that slide again, this will be and you know we are at the end of this lecture. Then I give you an explanation from two wonderful friends, Beatrice Colomino at Princeton and Mark Wigley in Columbia University. It all started with I'm a trained zoologist, stem cell biologist, the reductionistic school from a hardcore stem cell biologist. He was the last student of Max Delbrück, and you had to go deep and isolate, forget the environment, forget the surrounding, bring the stem cell into a petri dish and try to understand how does this stem cell really work. And I got interested in this cell biology and also in the evolution of the immune system. How are the cells protected or protect themselves? And it needs it's not very often that a scientist, at that time I was already a kind of an established scientist, changes direction. If you come up with an observation, you have two choices with an unusual observation. You have two choices to follow up. You ignore it because you see so many funny and different and weird things in your scientific life, or you think about it, and then you change the direction you go. And in the early days, a student came in my lab, Sebastian Fraune, and Sebastian is now a professor in University of Düsseldorf. And he came from the microbiology department. I'm sorry, running nose all the time. He was a trained microbiologist, and he wanted to do a PhD with me. And so I thought about it, and I said, Sebastian, there is a new world coming up. And the new world, means sequencing and technologies. And there's a saying that microbes are everywhere. Can we do sequencing? We sequence different species of Hydra species. And what you see on the left side is a phylogenetic tree of different species of Hydra. And on the right side, you see the cake. The colors of the cake is the contribution or the composition of the different microbial taxa. Sebastian did that, came to me, and then he came up, that is the second paper then, but in an earlier paper, also in PNAS in 2007, Sebastian showed me that all the different species have a very distinct and stable microbiome. He came in the office, and I remember, and I said, I don't believe you a single word, that's impossible. Because the animals live with me, I'm not a field biologist, they live with me in petri dishes, in glass dishes. I'll show you later how we keep them, and they're fed artificial food, artemia. Every species gets the same water and food and plastic environments. And they travel with me in my career from Germany to California to East Germany to many places. They always were with me. How can they have different microbes happens if they get for 20 years the same food and the same water and everything is artificial? I told him, go out in the wild. It was at that time I was already in Kiel, and go to some ponds and find the animals in the wild. I had no idea if they are even existing there. So Sebastian did that, went to the lakes in Plön, came back, we resequenced, and then he found by and large the same thing. So the 20-year-old cultures have by and large the same microbiome maintained or conserved as the wild animal. Ooh. We wrote a manuscript, 2 authors at that time, and sent it in to a prestigious journal. And the reviewers came during the reviewing process and said, this is great. Can we use the data? This will get published, but it will take a few months. I want to have the data for now. I'm writing a very visible review, and I want your allowance that I can use the data. I went back to Sebastian and told him, wow, this is something, we were out of the field. This is something really meaningful. And so from that time on, I decided to study that stable association between a microbiome and a number of different host species. For the very simple reason, if that is true, that you can take my chair there. You don't have to stand in the back. There's
Come on up, one person. Come on.
Okay. If they are stable associated for such a long time, the host must somehow do something to keep that microbiome in its or on its body, and hosts of other species must have other means, in a similar manner, of having a different microbiome. There is selection and evolution. There must be a reason behind that, why this species has this microbiome and the other one has this species. It's a genetic trait. And I decided to follow that up ever since then. And nowadays we know, and CES has published extensively on that issue as well in insects and on review papers. And this is just a very recent publication from my institution where a young postdoc, Malte Rühlemann, did that in hominids, and there's André Möller and others which had a science paper on the hominid microbiome. So different great apes, including Homo sapiens, have also their distinct microbiome, and you find no papers on fishes. And so this seems to be true. By and large, we call that a core microbiome. You can identify from a piece of feces, this is Homo sapiens, and this is orangutan, and this is whatever chimpanzee. It's a core microbiome. The rest of the microbes in your body vary greatly, depending where you are. My microbiome now is different since I'm here in this wonderful food here in Penn State. I will be on Sunday in Tokyo, or Monday in Tokyo. Will be the microbiome will change a bit. Okay, so this is the migrating microbe. Margaret calls it the tourists which are coming and going. If that is true, what I said, then the question is, why do hosts maintain the species-specific microbiota? And there is one currency in nature, and that is very costly. If you do something, you invest energy, ATPs, all kinds of chemical costs are coming up. So you better think about, why do you invest in that? And if animals or nature invests in something which is costly, there must be a reason behind. What is the function of the microbiota? And so with that thinking and with support of CES and others, we have founded a research center, Origin and Function of Metaorganisms. This is a new building, and my office is somewhere in the neighborhood here. And what we think is that it is time, and I'm very happy and feel very welcome here, that we have to give up the thinking which was in biology and in medicine predominating for 150 years or older. The focus was exclusively on the host. And it was not seen, it could not be seen, I think, that the host is closely associated with a complex menagerie of prokaryotes, eukaryotes, fungi, viruses, phages, all kinds of different organisms. But the host is not only associated with that, it's deeply integrated and actually inseparable. And you cannot separate the parts from each other. You can make germ-free animals. You cannot make, Erika told me that where she's sitting, you cannot make germ-free cows, which is a very interesting point for a very obvious reason. But all these germ-free animals are sick. And I could show you the anatomy of a germ-free mouse, and you would say, oh my God, how differences are in the organ sizes, and the length of the intestine, and all kinds of different things. So this is integrated into one association which is multi-organismic. That's what we call a metaorganism, or synonymously a holobiont. And this is very dynamic. Microbes can go and come in. The host doesn't change much. But all organisms are integrated into an environment. The environment is part of that metaorganismic concept. And if there is anything which allows you to adapt to new situations, it's a dynamic of the metaorganism. Viewing organisms and their associated microbiota as a single unit of biological organization that functions rather together than as they are not separate entities. That challenges, of course, traditional concepts of individuality in biology. One aspect which frustrates many people, also in the general public, is that everything is getting so complicated. And when we have discovered something, we realize, oh no, that's not the end of the search. It's getting so much more complex. My answer is, we have to accept complexity to understand fundamental problems. It's the contrast to the reductionistic approach. But we have to understand the complexity with the help of reductionistic thinking, because we want to go back and we want to understand the mechanisms, the causal relationships. Why and how does that really work in these multi-organismic associations? And that you cannot do with humans. You can do it with mouse, but it has limitations. You can do it with complex invertebrates and invertebrates, but it has complications. The best role model or model system would be a host with a rapid life cycle, established genetic tools, naturally microbiota, which is fully characterized and which you can culture. This is the animal I'm working all my life. I have never touched anything else. I got introduced to this animal by my teacher and PhD advisor, Charlie David, in a reductionistic manner, because Hydra was the better phage. In the school of Max Delbrück and others, the goal was to understand the fundamental rules of life. And they thought the phage is the good system for doing that because it's so simple. They invented molecular biology, but they failed to get to the fundamental rules of life. And the next best model was in the 70s, Hydra, by physicists. Alan Turing, in his book, The Chemical Basis of Morphogenesis, mentions Hydra as a theoretical model, how to understand how to make a top and how to make a bottom. It was not an animal. My PhD advisor was proud that he said, no, this is just a model to understand pattern formation. It's a great model. That's how we keep them. That's how we feed them. And that's how they look like. Very simple, only two cell layers, an ectoderm and an endoderm. They have forgotten, or they have not yet learned to make a mesoderm. And they have a very well worked out molecular biology of the pattern formation with different labs in the world. There is classical Wnt signaling pathways, there's Hox genes, there is beta-catenines. The whole molecular toolkit was invented by creatures similar to that, early emerging metazoans. And then it gets more complicated, more gene families, more numbers of genes, et cetera, et cetera. But the basic toolkit I would say, of everything of life was invented at the early days, about 560 million years ago. That's great. But there is an architecture of invisibility or invisible. That came only very late because I had no idea until the sequencing came that these animals are full of microbes. We can make, of course, visible now. These are genetically modified colonizers, which have a GFP integrated. We can make different bacteria, different colors, red, yellow, greenish. They live outside the organism. You have the mucosa in your gut, which is a very valuable barrier. They do, for some reason which you don't understand, they produce mucins, the epithelial cells. The mucins are getting secreted outside, and they have a mucosa, which we call glycocalyx, outside, where the different microbes sit. This is great, because the microbes are sitting outside. We just can pick them, and we can transplant back. We don't have to go anywhere inside. Inside is just a gastric cavity where actually microbes have a bad future because they are taken up and eaten. They sit outside in a layer which we have discovered very late by complex electron microscopy, cryofixation tissues. It's a very watery tissue, so it's not easy to work with. And in this layer, the microbes are laying. You can see them now with electron microscopy, and as I said, you can also visualize them. We had the sequence data, but now we could see them. And that was motivation. What are they doing? How do they communicate with the host, but also with each other? There are hundreds of different genotypes in this bacterial kingdom, which are colonizing this species and different one in other species. You don't have to read all that, but we can completely manipulate the host. We can knock out genes. We can ablate a cell population. We can do the whole toolkit of molecular biology, but we can do the same with the bacteria. We can genetically manipulate them. We have all the bacteria in culture, so we can really get to the interaction between the host and the bacteria in a living system. A model is a lie that helps you to understand the truth. I think this is very true. I'm working not on a lie, but I'm working on a model, and I'm very well aware that it takes a long time and complicated pathways to translate that into human research. I'm often invited by medical associations in meetings and give talk about Hydra. Because what I can do is I can deliver concepts. And nature is not evolving too many things de novo. Louis Wolpert said nature in evolution is tinkering with existing toolkits. So what we find here may be a little bit more modified in another organism, but the concept probably is true. And that holds true for all the observations we have done in the last decades. For this event today, I just want to concentrate on three aspects. So we have worked on many different aspects of the interaction here. I want to go on development, behavior, and the neural circuit assembly in embryos. Before doing that, summarizing, because I know there's amphibian people here, and amphibians are great models in a way that they show us the importance of the microbiome, because the frogs and the salamanders get sick. Why do they get sick? Because they get infected by fungi. Why does Hydra get sick when it gets infected by fungi? It only gets sick if it's germ-free. A normal animal never gets infected by fungi. But if you remove the bacteria, the animal gets killed. And that is true for many of us. And second, basic physiology in your body is driven and controlled by microbes. You have your spontaneous intestinal contractions. And if everything is fine, you don't care. Actually, you don't know. But sometimes you get pain. And Parkinson patients get pain, obstipation, irregularity of the contractions 10 years before they have the normal Parkinson symptoms. So what we have shown in Hydra is that they have regular contractions, but if you remove the bacteria, the contractions drastically go down and they become irregular. So even basic features of animal physiology are heavily influenced by the co-evolved bacteria and microbes. So the first, these are short chapters. The first chapter is not published yet. You can find most of the stuff in this bioarchive preprint. I find it super exciting, and it's done by a wonderful former PhD student and now a senior postdoc. Microbes affect stem cell decision. A lesson for the junior generation here. To learn new things, you have to read old books. We forget that. This was a mentor of mine, Pierre Tardin, a zoologist at the University of Zurich. And about in a science paper in 1982, I know that some of you were not born at that time, Israeli research team came up with a paper and claimed that in bacteria-free hydra, they cannot bud anymore. This is this proliferation by producing tissue extrusions, and then this is a bud, and then a bud grows, and then a bud falls off and makes a new polyp. So they say that there's a budding factor which is provided somehow, obviously by bacteria, because if you keep them bacteria-free, then they cannot bud anymore. You can imagine, I read old books, and the first thing I suggested to Sebastian, can we just somehow reproduce that, or is it true? And we couldn't. And there's nothing known, and there's no budding factor, and actually the whole story sounds very strange. Why should bacteria produce something which then an animal host needs for an essential part of the development? And then a young Chinese graduate student came in the lab, and I asked him pick up the project. And the difference to the former times was that you need a long vision, and we want to have long-term germ-free animals, not this 2 weeks after antibiotic treatment and then look for effects. And so Jinru was able to keep germ-free animals for up to 3 years and longer. Keeping that in the early days was meaning every single day, sterile feeding, sterile washing the animals. No vacation, no Christmas. He was every day in the lab. And he did not want to give that in other hands. He was very successful. He has now a wonderful position in our institution, in a medical department. And what Jinru observed is, so this is wild type, WT and CV is conventionalized. These were germ-free, and they were brought back the normal bacteria. And this is germ-free, and this is germ-free non-budding. And until before Jinru came, we were studying this animal versus this animal, and we couldn't find any difference. But if you keep them for a bit longer, you don't have to wait for 3 years, just 4 weeks or 6 weeks. You see that the animals get very weird. They get larger, they get fatter, they have more pigments inside, and they form ectopic structures. Their development is screwed up, and they stop budding. You can rescue that by bringing back the bacteria. And if you bring back the bacteria in these germ-free animals, they get a phenotype I have never seen before. They burst with buds. It is as if the bacteria which were brought back provide something or remove something which hindered in the early situation, the germ-free situation, which hindered the budding. And now it's just enormously low. This is a long-term study. I think it's the longest study before we have the original paper out, which I hope to do in 26 in a high visible journal. But I needed money to keep these projects going. So I told Jinru, we had an evaluation meeting, and with the referees from the German Science Foundation, and I said, don't worry, bring the living animals to the evaluation, to the referees, and let them see the animals. And that worked. And so we got the grant, and so we could continue to working on that, because that is absolutely astonishing. Animals are kind of, I will come back to that term later, architects of body plans. So how does the microbiome affect budding? Sorry that we have to switch now, and we go to high state of molecular biology, and we do what everybody does these days, single cell analysis. You have seen these atlases many times. So we have made single cells from all the populations of the body, of the animal, and then, but then you can apply clever bioinformatic. And if you apply clever bioinformatic, you can actually follow the trajectory of a stem cell, which always sits in the middle of this cloud here. So these are, for example, endodermal stem cells. So these are the endoderm cells, and these are terminally differentiated cells then of the endoderm. And this is the ectoderm, and the stem cells defined by their transcription signature. And then the signature changes, of course, if the cell goes into a more terminally differentiated pathway and finally ends up at a highly specialized ectodermal foot cell. So you can follow the trajectory from a stem cell to the terminally differentiated cell. You can do that for all the cell types. In addition, you can add another parameter, which is pseudotime. And you can ask, how long does it take to stay in that stage? And you see that the stem cell compartments have a very low pseudotime, but the differentiated cells have of course, a much longer pseudotime needed for doing this differentiation pathway. Back to the question, what is wrong in these germ-free animals? Superficially, or actually cellularly, at the phenotype level, you see this is a normal animal, and this is a germ-free animal, short time, and this is a germ-free animal, non-budding. You see a lot of brown spots along the body column. Oh my God, these brown spots are foot-specific cells. They express a peroxidase gene, where we have markers for, and we know this is very specific for the foot, but it never should be along the body column. And these are cells which are in this germ-free situation making the wrong decision to ending up at the wrong place in the body. Now it's getting a bit fancy, but I help you when I give you the message of these figures. So if you have the signature of the single cell analysis and the pseudotime, you can follow, just concentrate at the moment on this wonderful, colorful panel here. This is the wild type or the conventionalized animals. And what you have here on the axis is thousands of the genes which are identified by the single cell sequencing events. Thousands. Every single line actually is an individual gene, and I've highlighted some of them here because I come back to them. If you look on the left panel, you see this is the stem cells, and the stem cells go in our analysis through a phase of transition, and they then end up in the terminal differentiation, in our case here, in head-specific cells, and head-specific cells have head-specific genes. And in transition, they change from a stem cell-specific pattern in this terminal differentiated pattern. Look on the right side. What happens if you remove the bacteria? They cannot make the transition. There is no stem cell-specific compartment. Everything looks more or less terminally differentiated, and the gray bars, which you have to see here, are actually then genes which are not expressed at all. We find all the genes which you would expect. There's Wnt genes, there's FG1s, there is brachyury, there are Hox genes, but the genes which are needed for the stem cell compartments are not expressed, downregulated, whereas other genes, for this unknown gene, GAPR1, which is obviously very early on necessary, is also downregulated. This is needed for making a normal morphogenesis and budding, and this is what goes wrong in germ-free animals. If you add back the bacteria, as I showed you before, you can reverse the gene expression pattern. And then you go from this pattern to a re-expression of the normal pattern. Fascinating, complicated. You trust the postdoc that the bioinformatics really works, and you better do an independent experiment to prove that. And the independent experiment is always old-fashioned. Just do regular sequencing and transcriptome sequencing and see what's really going on. And that is a bulk RNA-seq experiment, so not single cell. And what you see here is just the typical patterns of genes expressed. This is the same genes what I showed you before, but now in a different order. And this is the gene expression in germ-free, non-budding animals. Most of them are downregulated because they don't do any of these continuous self-proliferation, budding, terminal differentiation, stem cell transition thing anymore. This is completely independent, less resolution. That's why we went to single cell, but then we looked back to this old data and said, oh yes, it's very nicely confirming what we find. And we choose for an example, we choose one of the genes which is downregulated, which we also detected earlier, GAPR1, and you can see this gene is expressed right there. This is HCR, so hybridization chain reaction, so very sharp pictures, and you see it's just there. You even can see the single transcript molecules where the new buds are coming out. And we did a control experiment and knocked down the gene. And if you knock down the gene, this GAPR1, the animals cannot or are very defective in budding. So that is just to prove that our overall view, most of the genes are downregulated, particular early genes, which are necessary for the transition of a stem cell to a differentiated cell. If you knock it out, then the polyps cannot bud anymore. Fascinating again. But no, not the end of the story, because now you have to ask, how does this work? How can microbes, which are living outside in the glycocalyx, have such a deep influence on transcription pattern of the host? That can be indirect, which we then have difficult to understand, or maybe direct. There's another novel technology where you can analyze that, and it is ATAC sequencing, assay for transposase accessible chromatin. And what you do is you again do a single cell experiment, but then you sequence with this type of technology, you cut out the open chromatin regions. This is where the music plays, where transcription factors can bind to and activate genes, et cetera, et cetera. So you cut out all these pieces here from single cells. And then in addition, you add a primer to the sequencing kit, which are specific for transcription factors. And then you can analyze this sequence here from the host, plus the transcription factor binding sites, which you just take from a data bank. We took about 1,800, something like that. Then you can directly see if there is in the open chromatin a signature which is different in germ-free versus normal animals. And sure it is. This is another single-cell atlas, but it is now the chromatin accessibility landscape of different cell types. These are the endodermal cells, which are the stem cells here, which I shown before, but this is not gene transcription. This is not genome architecture. Means open chromatin regions, of course, change in different cell types because a blood cell expresses other genes than a muscle cell. So, and that is due to the differences in the open chromatin. So different cell types have different open chromatin landscapes, and the germ-free animals are different from the controls. That was terrific, because now we show that microbes directly influence the chromatin landscape of the host, and that fits very well, which I strongly believe that the music is played by epigenetic means. The microbes produce something which affects the epigenetic machinery of the host, and that changes the chromatin landscape. And you can map that or visualize that again. This is now quantitative. These are different chromatin. These are not genes anymore, but these are different chromatin pieces of the different cell types. And what you see here, this is the ectoderm and this is the endoderm. And in the non-budding animals which don't bud, the chromatin landscape is very closed in contrast to the controls, and that is true for the ectoderm versus the endoderm. That fits very well with our transcript data because many genes are downregulated because the chromatin is closed and the transcription factors cannot get to the binding sites where they then activate or deactivate certain genes. So I think, this is at the moment, we are now, the last piece of this paper is gene regulatory networks in the presence or absence of microbes. I think that there's a substantial influence of microbiota on host genome architecture, the microbiome, a central layer of gene regulation, still question mark. In summary of this first chapter, microbes are not just communities, they act as architects, if you think about that, of our bodies, are essential players in animal development itself. And during long-term absence of the microbes, factors accumulate in the hydra host that are inhibitory for budding. We can discuss that we have very recent wonderful data. We don't think that the microbes, as in the Science 82 paper, provide an active budding factor. What they do is scavenger theory. They take away waste all the time. It's also in your body. But if the microbes are not there anymore, the waste accumulates. And if the waste accumulates in State Park, the streets are very ugly and it's not very healthy. And that same is true in our bodies. Microbes are perfect waste workers. This dependence of this asexual reproduction of the host on its bacteria may be the selective force that has established this ancient phylosymbiotic relationship. You cannot separate the microbes from the host. The host is not able to survive for a long time. Fundamental influence of microbes is, of course, also seen in other organisms, in mammals, architecture of multiple organs, intestine, hair, skin, lungs, in fish, etc. I'm a developmental biologist. I was president of the Developmental Biology Society for some time, and I try to convince the developmental biologists of this time that development is really a complex interplay between environmental-microbial factors and the host genetic. I'm not finished with this mission yet. The second chapter, microbes control behavior. This is fascinating. And the paper was published in 23 in Current Biology, and it's done by another gifted graduate student, Christoph Dietz. Christoph is now a postdoc at the Francis Crick Institute in London, and he studied the eating behavior, the mouse opening behavior of the animals. And this is my community ignores that the animals have a behavior. But there's old data, read old books, 1955 from Bill Loomis, which shows a number of different behaviors. And we thought that this behavior is not independent of the microbial colonizers, but the microbial colonizers and the host are driving together, making this stereotypic behavior. And one of the stereotypic behaviors is this mouse opening, which you can see here in this animation very, very well. But you can now study very deeply, and that is what Christoph really did. And first of all, what we have to do is you have to understand the neuronal context, and on top of that, then lay the microbial context. So you look here on two populations of transgenic neurons, blue ones and red ones. Don't worry about the details, but here is the mouse. And the mouse opens regularly to eat, of course, and we have calcium imaging constructs in the neurons which are surrounding the mouse, a very specific subpopulation. And you can see this is now live. Calcium imaging means firing of neurons when the mouse is opening. And if you remove these neurons, which we can do by cell ablation experiments, then they cannot open the mouse anymore. That's the end of the story. You have then somehow to rescue them. So we have a tool to understand the neuronal context. We have worked out the whole nerve system of the animal. It has 7 distinct populations which work as neural circuitry, and different neural circuits are integrated with each other and manage the whole behavior in a very distinct manner. never talk about a primitive nervous net, which you find in all the existing textbooks. This is super sophisticated, and we still have not understood all the details, how signals are transmitted through all the body column, etc., etc. Now we are talking about an N6 population and an N4 population, which is throughout the body, and these nerve cells here regulate the eating behavior. And Christoph has shown that first the signal, which then allows the animals to open the mouth, comes to the nerve 6 cells, and then it goes to the nerve 4 cells, and at the end, then the mouth can be opened, and we have nerve subpopulation specific promoters which drive then the calcium imaging construct, etc., etc. So this is worked out very well. What has the microbes to do with them? You do that first in germ-free animals, and we had many different people in the lab. We always realized that germ-free animals are not as good in mouth opening as the normal animals. So then we studied that systematically. What we did is we monocolonized a germ-free animal with one bacterium, Corfibacter. This is where the Corfibacter sits around in the top part here, but there are also some bacteria. And Christoph came, it's one of these other aha events in your life, and which you never forget again, and which you cannot believe. And he said, Thomas, recolonized animals cannot open, monocolonized animals cannot open the mouth anymore. I said, what? This is the most essential thing. If you cannot open the mouth anymore, that's it. How can that happen? They cannot open the mouth. Germ-free animals are doing bad, but Corfibacter monocolonized only animals cannot open it anymore. How does that work? So we did genome mining of this bacterium and asked what is the molecular feature of this bacterium. And the molecular feature of this bacterium is among many other things, it produces tons of glutamate. And this triggered something because we know for a long time from Italian scientists in the 80s that the neurosystem in Hydra somehow has glutamate receptors. At that point, the previous researchers had to stop because they had not technology to go deeper. So we knew that in our N6 neurons, there is a glutamate receptor. And now this animal, which is monocolonized in the tissue, produces tons of glutamate. These tons of glutamate occupy completely the receptor. And the end is that this receptor here then leads to this control of the eating behavior. In our case, not activating the epithelial cells which open the mouth. If we check that chemically and add pure glutamate in contrast to other amino acids, etc., the same thing, of course. If you expose the animals with glutamate, they cannot open the mouth. How does that work in reality? This is very superficial, it's very artificial. Animals are never alone. Animals have a bacterial community. And if we add back some of the other companions of this Corfibacter, in particular, if we add back Duganella, the mouth opening behavior comes again. And why? Because genome mining has shown us that Undibacterium cannot make a single molecule of glutamate. And in a normal diverse microbiome, the glutamate produced by this guy is used up by this guy. And the host cells, the glutamate receptor, never sees the glutamate. For me, that is the message that you need a diverse microbiome. And if you reduce the diversity, this is dysbiosis, and it may have unforeseen consequences. We never expected that a monocolonized animal has these drastic problems here. And if you rewild the microbiome, then the problem is gone. Last chapter, a short chapter, neural circuit assembly in embryos, was published earlier this year by Christopher Nowak, another graduate student. He's now postdoc in the University of Vienna. Every graduate student in my lab gets a challenging task. Yes, he or she has to develop something really new. And Christopher came and I said, it's great. Let's look now in the real development of animals. That's embryogenesis and not these adult polyps which bud. Until that time, nobody could quantitatively work with embryos. Christopher, this was of difficult. You have a Christoph in the lab and a Christopher in the lab. But Christopher managed. And what we were interested in is, this is how your brain developed some years ago. And it comes from neural circuits which are getting organized and which have already some functions. And these neural circuits then also get integrated in the human case or in vertebrate case in a very complex brain. But evolutionary, of course, you can go back the way, and then you start from an individual neuron, and then individual neurons that go to these circuits here, and then circuits get different signatures. That's why we have, along the animal, if you remember, we have an N4 population, we have an N6 population, and these circuits have different functions, gene expression, location in the body, and together they make the nerve system. So I asked Christopher, do that in embryos, and so this is the whole embryogenesis. It takes about 12 hours, and you see here a movie where this wonderful little polyp comes out. This is the end result. This is a transgenic animal, then has some transgenic neurons. If you see like that, it's just wonderful. Students don't appreciate that. They just, I have made transgenic animals, and I have no hatchlings, but this is wonderful. And so the hatchling comes out, and then you can study that. And what Christopher was studying, the assembly of the nerve 4 circuitry during embryogenesis. And again, you have to survive for 2 minutes of neurobiology before we come to the microbes. So this is calcium imaging in individual embryos. So he localized the neurons, or they localized by itself because they are shining. When they are active, they're firing, and they give a calcium signal. And what you should see here is, by and large, that from single scattered neurons, with time, you get to communities. And this is now this is an N4 circuit here, which was made during embryogenesis. That was very essential to show, because previously in all textbooks still, hopefully not next year anymore, but think that ancestral neural circuits are randomly connected nerve cells. No. And you can visualize that here very nicely. And you see from an initial beginning, there are some neurons which never make it to that net here. And then you have this is the N4 population here. And there's some pruning going on. And that is just one out of at least 7 different populations which are developing, done in real time, in real embryos. Now, of course, I received this wonderful medal only when I talk about microbes. So I have to go now to the microbial part. And the microbial part is unbelievable, fascinating, and I could not believe it. But to be honest, when Christopher came to the lab, I said, you do all this neurobiology and embryo work now. That is great. But I have one dream. You repeat then the experiments with germ-free animals, which is more difficult because the germ-free animals are not as good as the normal animals. But at the end, before he got the job to Vienna, he fulfilled me my dream. First of all, I show you the complexity of the embryos. This is done by Alexander Klimovich, a senior postdoc in my lab. With a spinning microscope at the University of Vienna. What you should see is, this is the embryo here. And this is what we saw before. But I never had any idea, similar to the glycocalyx mucosa experience, that there is so much stuff. This is DNA. Some of the DNA probably is from dead cells. Not all of them are living cells. But there is a layer of a biotic environment surrounding the embryo. And you can even dig deeper, and this is done by Genro. You can now use specific probes, eukaryotic probe, or fungi probes, or just a DAPI. What you can see is, this is the same thing what I have shown you before. This is more or less what Alex has shown with his spinning microscopy. But you see here the red spots here, you see yellow spots, you see green spots. Oh my God, that's the whole microbiome, including mucobiome, surrounding the embryo. What does it do? I will show you, then I'm closing, that in the absence of these signals, by making germ-free embryos and following them with time, we show that the nerve network is significantly impaired or not really functioning. First of all, this is the trouble. The germ-free animals don't hatch as efficient as normal animals. This is shown in this graph. Second, the number of neurons in the different developmental stages in the hatchlings in the germ-free animals is always much lower than the controls, which are the recolonized, this is the conventionalized, and the wild-type embryos. So lower numbers of neurons, and most significantly, a neuron starts in development and has two processes, two neurites. And then the number, these are 3, these are 4, and these are 5 neurites here. The number of neurites increases during neurodevelopment. In the germ-free animal, they always stay with the initial situation. They have 2 neurons. In the control animals here, you see that the number of the 2 neurites are decreasing because most of the neurites, because neurons, the number of 2 neurites is decreasing because these animals, in the presence of the bacteria, get more neurites. What does that mean? That is functionally super important because you need a number of neurites to form a nerve net. And if you have only 2, then the possibility that you make a functioning nerve net is very limited. And that's what we showed. So this is the wild-type nerve net. Again, it's always with the nerve 4 population. This is the future mouse and the future head of the animal. This is the foot here. You see a wonderful early embryonic nerve net, and you can you get the same thing if you recolonize the animals with bacteria. But if you keep them germ-free, this is not a functioning nerve net, and the animals also don't do well. They get out of the shell, cannot do the pumping much, and they have not a good future at all. Message: neuronal development is not purely genetically determined, but is highly plastic and influenced by environmental factors such as the presence of the microbes. What is true for Hydra is true for man. We developed concepts in a beautiful paper by Lin Chao from UCLA, has shown some time ago, I think it was in Nature, yes, in 2020, that the same is true in mouse embryos, which are developing in a germ-free mother mouse, so missing maternal microbial derived metabolites. And what happens is that the neurons in these embryos, in these mouse embryos, which are in a germ-free mother, have much less protrusions than in the control situation. So same thing. If you want to have a fully functioning nerve net from Hydra to mammals, you need the presence of the microbes. And it's not, as I said yesterday, not this microbial species and cell, it's the microbial metabolites which matter. So I'm coming to the end. I've shown you that microbes modulate stem cell activity, that they influence behavior, and they also influence the neuronal architecture. In my last 1 minute, I devote to asking, why should you care about that as a scientist, which is not working on Hydra? And you should care about that because this has deep influence on how we think about evolution. We have to integrate in our evolutionary thinking and concepts and theories that microbes play a central role. You start, and you know that very well, that eukaryotic cells are a symbiotic event. The chloroplasts and the mitochondria are of bacterial origin, etc., etc. And so we have many, and I've tried to show you that some of the effects of the co-evolved microbiome on shaping the phenotype. This is getting a hot topic in evolutionary biology. And for one reason, it challenges the gene-centric view. The highly visible person who has invented the gene-centric view with numbers of very well-read books, good to read, has just published another book where he proposes this view. And I think it needs to be modified, at least, because microbial communities with their own genomes are equally integral to the evolutionary approach. The second reason why that matters is the more we understand how important the microbes are, the more we have to be worried about in our time, which we call the Anthropocene, that these essential partners of life are having not a place anymore in our body, in our world, in our soil. And this is worrisome because I tried to convince you how important they really are. Lifestyle diseases, the deterioration of our health, disturbed microbial networks. This is why we need an understanding before we can do something, but then we have to do something. Nothing in biology makes sense except in the light of the holobiont or the metaorganism. That's what I truly believe. And that is true not only for biology, but also for medicine. I promise you that at the end, we, the bacteria, my two friends, Beatrice Colomina and Mark Wigley, and just these days, actually in January 16 next year, their new book comes out. We are friends for a long time. We spent together a wonderful 1-year sabbatical at the Institute of Advanced Research in Berlin. They're architects and designers, and they think as you and I do. They think holistic, and they think architecture new in a very provocative and drastic manner. They are curators of the Biennale at the moment. These days they are in Milano, and think that even anything where humans are touching or living on or staying in is influenced by the microbial world. And so we have published a paper on the importance of the built environment microbiome and its impact on human health, together with Mark and Beatrice and the group from the CIFA think tank. We, the bacteria. With that, I thank, first of all, my team. I've mentioned all the people which were really involved in that. I thank the funding situations, and I thank you for the minute. Thank you so much.
Thomas, thank you for the thought, for the performance of how we are engaged in your work and how you present it. It's very refreshing. We're a little over time, but I think one of the best things about Thomas's presentations is they really encourage discussion. And so for those that need to leave, please go, but I'll take a couple extra minutes for questions so we can enjoy that part of Thomas's scholarship. Usually we start with a question from a trainee. So it looks like there might be one already in the back. Yes. Yeah.
Thank you so much for your talk. It was absolutely amazing. I want to go back to your second and your third chapter, and I was wondering if you ever looked at or considered looking at when you maintain multiple generations of the host, in this case the Hydra, within a state where the microbiome is either absent or in the state of dysbiosis, how does that affect host genetic variation? Because I'm assuming that that's going to be a strong selective pressure if it affects the developmental stage and the correct function of the neurons. So I was wondering about that.
Excellent question. Wonderful. I only can partially answer that. So we have not done, and we don't know yet. So the genomic architecture really, what's really going on in terms of variation. What we have done is we have done the chromatin landscape. That's what we know, and we know the gene expression patterns. They are drastically different. But how variable that is at a genetic level, we still don't know. It's a very good question. Yeah, thank you. These are clonal animals, right? So should not be too much variation, but I learned to accept surprises.
Yeah. Question here.
Yeah.
What happens if you infect a germ-free embryo with the microbiome from a different Hydra species?
Done. Thank you so much. Very good question. And they cannot colonize the animal. There's a reason we know that. This genetic we know, the host and also your body produces sets of thousands of antimicrobial peptides. And these peptides, they regulate the endogenous, the normal microbiome. So if you remove the bacteria from the host, it still produces this type of peptides. And if there are foreign bacteria are coming, they cannot colonize.
I was really fascinated by the eating control, eating behavior control. So do Hydra have sort of concept of satiety? Like, do they, if there's food there, are they just always eating it, or is there some
No.
control?
Published eLife paper last year. Same graduate student, Geetz et al., and we called it internal state or satiety. And after feeding, the animals normally are saturated and cannot open the mouth. It needs some time. So, and we know the neuronal cell types which are responsible for this internal state.
Hey, can I just follow up quickly?
Sure.
So you have glutamate
Mhm.
production. Is there GABA production?
There's also GABA.
And is it?
There's also
Functioning
dopamine. There's also dopamine.
And yeah, I just wanted to functioning similarly in humans where they're kind of opposing pathways and where
Yeah.
that's related to the eating behavior.
This we don't know really. The glutamate story is interesting because I think this is the evolutionary origin of appetite. Glutamate in our body is appetite. If you go in the supermarket and buy your processed breakfast, Marianne, in the plastic dishes, and you read the small things, you always find glutamate because it activates our appetite. So the glutamate story, but this is an interesting question which we have not yet fully understood. I have neurobiologists which are pushing me to understand this GABA, dopamine, etc. network. I would say if you want to do that, I'm happy to send you animals.
That's not something I'm doing.
Yeah. But what I want to say, I after so much time spending with these animals, I'm really convinced that they reveal the ancestral toolkit. It got a bit more complicated in our body, but the basic language is there. And language is always chemical. But the new thing is the chemistry can come either from the host or the bacteria.